Who am I? Well, who is anyone? I mean, I'm a water polo player, a robotics kid, a physics nerd, a peer counselor, and probably a lot of other things. That doesn't explain much as to who I am, though. When I think of what makes someone who they are, a list of adjectives doesn't just come to mind. I think about what drives them forward, how they respond to different pressures, what they choose to do with their free time, how they think, and so on. Those are a lot of things that would be really difficult for me to express in writing, so instead I'm going to use a sort of draw-my-life conversational way of explaining who I am. Except, I tried laying out my life from start to now, and there's just too much to cover here, even if I just isolated high school. Instead, I shall call this Draw My Summer Biomedical Research Internship I Did Before Starting Senior Year, because that's a much longer title, and I can cover all of that in a single video. So I'm not much of a bio person, but I decided that I wanted to give biomedical research a shot. Maybe I would really like it and find I was better suited for science than for engineering. I knew I loved robotics from all the time I spent on my FRC team, but I wasn't sure about science. My junior year of high school, I applied to a summer research internship program at the Children's Hospital of Los Angeles. And much to my surprise, I was selected for an interview. For my interview, I was selected for the internship among about a dozen other students. I was excited to get started, but also a bit nervous in the lab. Like I said, I'm not much of a bio person. The 2 weeks before the actual research began, our own Hell Weeks, as the program admins called it, served as a training period, where we would learn everything we needed to know about working in the lab. I used this period to learn a lot of bio that I had forgotten from school, such as what the heck is a plasmid and why are we putting it into bacteria. With the help of a friend a bit more well-versed in bio, I was able to figure it all out. While she was able to help me with all the conceptual biology stuff, there wasn't much she could do to help me with in terms of working in the lab. That was the scariest part, since I had never worked in a lab before. A few experiments for school here and there, yes, but not a full-blown actual lab. The idea of making a mistake was terrifying, especially knowing that Dr. B, the man training us, would tear any of us apart for forgetting a single fact about mitosis. As luck would have it, this fear of making mistakes made me more susceptible to missteps. Rushing to finish the training lab, I screwed up big time. I was supposed to create 2 samples to send through a gel electrophoresis, one of RNA and one of DNA. Instead of doing that, I made 2 samples of RNA. That meant that when the gel was imaged, nothing would show up. The thought of confessing my mistake to Dr. B paralyzed me for a moment, but I knew he would find out one way or another. I shuffled up to Dr. B trembling and confessed that I had just wasted RNA on the wrong sample. I braced myself for a scolding, and I was pleasantly surprised. Instead of tearing me to shreds, he put his hand on my shoulder and said, "It's okay. You can just have 2 negative controls, but this actually brings up a very important point." With the other intern's attention, he explained that failure is not inherently bad, and that in fact, mistakes are good. They teach us that we're not always going to do everything perfectly the first time, and that we need to change whatever it is that caused us to fail. That speech calmed my nerves more than anything that summer. After these grueling 2 weeks, I was assigned my lab partner and mentor, and the research began. I was excited to get started on our research as it involved a potential cure for diabetes. The actual cure would be way far down the road from where we're starting, but it was still pretty cool to be involved in that. The idea behind our experiment was pretty simple. There are 2 important cell types in the pancreas, beta cells and ductal cells. Beta cells produce insulin, while ductal cells do not produce insulin. We wanted to find out why. We hypothesized that while they both must have the same gene for insulin production, PDX1, it was being suppressed in ductal cells through DNA methylation, which is when a methyl group essentially says, "Nope, you aren't using this part of the DNA." If this was the case, maybe scientists further down the line could find a way to get rid of this methyl group and replace the beta cells lost in diabetes with ductal cells. Maybe. It took a while for my lab partner and I to get the cells we were planning to work with, so in the meantime we did a bit of a trial run with some mouse cells. By the time we actually got our cells, we were in for a bit of a time crunch. A bit of a very serious time crunch. At the end of a really long procedure for just isolating a bisulfite treating our DNA, we made a fatal mistake. We were at the very last step in the procedure. All we had to do was put the DNA samples through the centrifuge, where it would be washed off its filter and into its test tube. We did that, except that after several washes purifying the DNA, we didn't change out the test tubes that the DNA would be emptied into. That meant that we emptied our DNA into a tube filled with all of this junk that we had just spent over 2 hours separating our DNA from. Even though it would mean staying in the lab a bit later than we had hoped, we decided it would be best for the flow of our experiment if we just ran through the whole process one more time. We went home pretty late that night. We never made that same mistake again, though. Never again. Always double check. The time was finally coming when we had to present our findings at a science symposium. The problem was, we didn't find anything. By the end of our experiment, we had no DNA sequences. Instead, we had these little slips of paper back from the sequencing company that said, "We can't sequence this." So here we were, a few days before our presentation, with nothing to show for over a month of research. We looked to Dr. B for guidance, and he explained to us that this happens often in science. Things don't always work out perfectly, and oftentimes scientists will try to cover up these blunders by changing the premise of their experiments after the fact. That's not what our plan was, though. Dr. B suggested that we run one last PCR on our DNA. We could run this PCR through a gel electrophoresis, and it would show that we at least managed to clone the right gene. We would have something to show at the symposium. Sounds good. The problem was that after running the gel, we found that we had cloned a strand of DNA that was much shorter than we expected it to be. We weren't sure what we cloned. With only a few days before the symposium, I suggested, what if, after we explain our procedure, we pretend like it worked out perfectly. We'll say, without further ado, here are our results. And we'll wait a moment in silence, as though we're waiting for the next slide with our results. And then we'll say, "Oh right, it didn't work!" My lab partner loved the idea, and our mentor said that we could just go for it and beg forgiveness from Dr. B after the fact. With the strict 5-minute time limit, I was afraid that my lab partner and I would lose the audience. However, we didn't let that hinder the presentation. I wanted it to be interesting and engaging for everyone, not just the PhDs. We replaced expensive scientific words with layman's terms, and we were bold with our results. We transitioned into our results with a promise of DNA sequences, only to exclaim, "Oh right, it didn't work!" In that moment, I had full confidence in myself and my lab partner, knowing we were owning up to our results. Our audience didn't just like the twist, they loved it. We had a crowd laughing with us in a situation I wouldn't have expected at a science symposium. We may not have conjured the next cure for diabetes, but I'm sure our audience enjoyed our presentation for its charm and honesty of failure. It's easy to hide our mistakes by only being honest about what worked, so it's refreshing to hear that something didn't work. Only by identifying our shortcomings can we overcome them.